we looked at this as an idea of how they could possibly work with their environment, work with what they have available. So they had to make do with what's around them. And evolutionary microbiologists agree that, iron and hydrogen and sulfur, that was all pretty abundant billions of years ago, no oxygen. And so the microbes had to evolve and adapt to their environment and come up with a way to get some ATP generated. And it's a universal truth that if you can create a proton gradient across a membrane, you can get ATP. You're doing it right now, especially if you had a good lunch where your mitochondria are doing that full tilt and creating a lot of ATP for you. And so the goal for the microbe is, basically they had the iron and hydrogen available, but that's really not what they were going for. They're like, I just got to cobble something together in order to get ATP. So my goal is I need a proton gradient. And so they had to work with what they had. And that's what you see here. If you look at the iron sulfide and the hydrogen sulfide reacting, and really they didn't care so much about this product, the FeS2. What they really cared about was the H2, because they knew the rules of the game. If they can get and strip electrons off of that in order to facilitate the reaction inside, one of the products are protons. And if you do that reaction over and over and over and over and over, you'll get a nice concentration of protons on the outside of the cell, not as many protons inside the cell. Well, boom, there's your proton gradient. So what they're really trying to do here is assess their environment, figure out what they have, and then go, okay, I want some ATP. I need a proton gradient for that. How can I do that? And that's essentially what they're trying to do here with this hydrogenase, stripping electrons off. This is helping facilitate that internal reaction that gets you the hydrogen sulfide, that then facilitates this reaction, and it just keeps going round and round and round and round and round and round so that you get a buildup of protons. Make sense? Okay. So how many people have taken BI, what is it, Intro to Cell and Molecular, BI 218? Oh, lovely. So this should not be brand new to you then. I'm guessing they talk about that a little bit in that class. And hopefully you're getting it in some other classes too, because it really is a universal thing. It's not microbes, microbes aren't the only ones doing this kind of thing. Living organisms create proton gradients. That's a good thing. Okay. What we need to think about now, I'm going to get into the next slide set. And, whoops. We need to think a little bit more about the process of evolution. And how many people have taken Intro to Evolution? Okay, a little bit less, but some. All right. So when you take a standard Intro to Evolution class, they will talk about mutations. And that mutations are driving the evolutionary process, or one of the mechanisms. Well, yes, but that's not the only thing to think about. So when you think about the word mutation, it just means a change in the nucleotide sequence of the DNA, all right? And so this idea that you have mutations happening, it could be because when the microbe decides to go through reproduction, when it was copying its DNA, it made a mistake. Or it could be that they got exposed to ultraviolet radiation, and that, as we know, UV light's not good for us. It's not good for microbes either. And so that could cause some DNA damage. That's what it does to you. If you go and lay out on the beach, you get a suntan. The suntan is your body's way of trying to prevent DNA damage. And then there's, like, in the lab, very often if we want to try to create a mutant quickly and we're not too fussy about where the mutation happened, we'll hit it with UV light. That's a very standard technique. You can add chemicals, there's all kinds of other things that could happen where you get mutation events. And then if you think about this idea that, okay, well, let's say there is a mutation in the DNA, and maybe that mutation creates a situation where the microbe can better adapt to their environment that they're currently in. And so you would say, okay, adaptive mutations then improve the fitness of the microbe in that particular environment. So you have to think about that particular environment. Because just because they're well adapted to that doesn't mean they're this perfect organism and they can live anywhere. Okay, so you always have to think about the context of what is that microbe experiencing, and is that going to be an adaptive mutation or not? The other thing I want you to think about is that when you get into the microbial realm, the prokaryotic cell type, they are genetically haploid. We're diploid. We have 2 copies of our DNA, one from your mom, one from your dad. But they only have one, okay? 1 copy of each gene. So that means if they get a mutation, it could be pretty disastrous. All right. And so microbes are pretty good at repairing DNA damage. It's pretty critical for them. But you can still get some funky things going on. Some microbes routinely will go through what they call gene duplication, which is, I still myself don't fully understand how this happens, but basically you'll get another copy of the gene. And they see it happening during reproduction when that cell decides to reproduce. And sometimes that gene gets maintained as a second copy. Sometimes they see microbes get that second copy of the gene, and then it almost like goes through this heightened mutation events going on where I picture the microbe like tinkering, like he's a carpenter or something, and he's trying to tinker with it. Like, what could I do with this to create a situation that works really good for me? And so it's an interesting thing. I don't fully understand it, but you have this gene duplication that could help with, if there is a mutation, maybe that is something that could help them, or it could create a new gene that helps in some way. What I'm going to focus on greatly, though, is looking at horizontal gene transfer. And so this is something that the microbial world does. And we're going to get into that more in the next slide set. But this is the idea that you have genes getting transferred to cells that live in the same environment. So neighboring cells can switch and swap DNA. And that's when that was initially discovered, it was huge news and almost met with disbelief. It's like, how, what? But they really can do this. And it has led to some very interesting traits. It's also led to some scary issues for us, which we'll talk about. Another thing to think about, which often I think might get overlooked, is that microbes are not afraid to purge DNA. And so they're so tiny, the amount of DNA that's packed into their cell is pretty enormous. It's a lot of upkeep to maintain that DNA, all those genes. So if they're in a habitat and they're like, you know what, this gene, it's just been sitting idle for decades, so to speak. I don't need it. I'm going to purge it because it's less upkeep for me, less energy expenditure for me. All right. So microbes in particular and viruses, which aren't really considered living entities, but microbes are pretty good at purging DNA that's no longer useful for them in that particular habitat. So that is another avenue that I think often gets overlooked in a general evolution class. And this can be super adaptive and really good if you are, for example, a parasite where you live long-term with your host. You make the host do pretty much everything for you, and you just kind of siphon off nutrients from the host. And so parasites can be like that, obligate symbionts that can't exist on their own anymore because they purged so much of their DNA. They don't have the tools to do that free living experience. They have to be with their partner, their host. And so, but it's still effective for them. It keeps them very efficient, and they rely on their host for some of the loss that they have purged out from their cell. So in order to study all this, you get into the idea of phylogeny, which is just basically looking at how are organisms related to each other, you know, the phylogenetic tree, the tree of life. And right now, the best way that they see to do this, to try to figure out relatedness among species and speciation and diversification, is to look at nucleotide sequencing. But they had to figure out which stretch of nucleotides should they sequence and zoom in on. And so you might hear the word chronometer or molecular clock. That's often what they refer to. And so they had to pick a genes or something that was effective at measuring evolutionary change in the right way. And every organism has that, and it doesn't change too fast. It was a lot of effort to come up with this idea. But basically, what's currently being used is the so-called small subunit ribosomal RNA genes. And they abbreviate that down to SSU, ribosomal RNA, rRNA. So they take the stretch of DNA that codes for ribosomal RNA, and they use that as a chronometer or a molecular clock. So that's what they're comparing amongst all the different organisms as they're trying to fill out a phylogenetic tree. So with microbes, that can work. And when they do that, just be aware that microbes or the prokaryotic cell type have the so-called 16S ribosomal RNA, and the eukaryotic cell type have the 18S. So there's a little bit different, but basically they're looking at the gene that codes for the ribosomal RNA. Because it's pretty steady, it's constant, it's conserved, which means it doesn't change very long, very quickly, and it has enough bases to have some good data to look at and see the changes. So this was a huge breakthrough in looking at phylogeny and trying to create phylogenetic trees. And so remember, you don't have to write all this down because this is all available. So don't feel like you have to write verbatim what's on the slide. This is now considered pretty standard. There's all kinds of businesses out there that will do this for you. If you just send them some DNA, they will do all the sequencing and they'll create the phylogenetic trees and all of that. And so it is something that's routinely done. The prices come down quite a bit, thankfully. But of course, if you want really good detail, like if you say, I've given you this DNA, I want you to tell me, you know, all the way down to the species level, what we have. And if you go all the way to the species level, it's more money. If you say, oh, just take it to the family or to the genus or class, then it's not as much money. But the kind of rule of thumb that I'd like you to know is that they say if it's 95% or greater in terms of it, you know, they're identical. If you're comparing to 16S ribosomal RNA genes from two different organisms, and you're saying, are these the same organism or are they different? The rule of thumb is 95% or greater means you have the same species. That's what I'd like you to know. Now I will be asking about that. So that's the current rule of thumb. Be aware that could change. It depends on with more technology, more thought over this. Sometimes you get some shifting going on, but that's the current rule of thumb. So I already showed you this idea with Dr. Woese. He was the one who came up with this idea. Let's zoom in on the ribosomal RNA gene and compare that amongst all different samples. And that's how he was able to actually say, you know what, there's a whole group of organisms that are different enough. They don't really belong. They used to belong into the domain bacteria. And he said, nope, they deserve their own domain. They're different enough. And so he is credited with that huge breakthrough in this idea of using the ribosomal RNA. And this is a partial phylogenetic tree, very non-detailed, but the three domains of life are listed up there. And I couldn't get the circle to move because of they wouldn't let me unlock that. But basically that little circle should be more like right here. So there's the base of your tree. Luca, last universal common ancestor. And then you can see the diversification, speciation coming off of that. And so here in the blue with the bacteria, you even see that they listed out the mitochondria, the chloroplast. So again, trying to emphasize the idea that they were once free living organisms. It's just now they've gone endosymbiotic and have evolved into what we call the mitochondria and the chloroplast. Another thing here, I said that the archaea were once lumped in with the bacteria, but when they really did this ribosomal RNA comparisons, they actually found something interesting. So if you look at this, the way the artist has drawn this tree, okay, you have the root down at the base, and then you can see, the blue line going off to the left, and then this one coming off to the right. And it's the one off to the right where you see the archaea branching off of it. So what that really means, and the artist is correct on how they drew this, the archaea turn out to be more closely related to the eukarya than they are to the bacteria domain, which is not what they thought at all. So this was a huge surprise, and one that still is very interesting. And so the archaea actually have more in common with the eukarya. They branched off of that line of descent rather than coming off of the bacterial domain there. So, and then I've showed you this tree of life. So there's all kinds of tree of life that can be drawn, depends what you're focusing in on. This one is again showing how much of the life is prokaryotic and microbial. So now we need to think about, let's see. So in lab, it won't be until next Thursday. Next Thursday, when you look at your experimental results, you're going to be seeing some morphology. And so very often, when you look at different organisms, you can look at, well, what do they look like? A grizzly bear looks different than a salamander. With microbes, it's not quite so clear cut because you can have microbes that look very, very similar to the naked eye, and they can be extremely hard to tell apart. So that's a little frustrating. But next Thursday, we'll get you starting to look at colonies on a plate, the microbes on the plate, and there are some very good things you can pick up from just looking at how microbes grow on a plate, under medium. And so we'll start training your eye to that. But here's some other ideas that they can look at to try to tell if organisms are the same or different, especially in the microbial realm. And so we'll look at some of these, like, is it normal or not? What kinds of physiology does it do? And we won't do this, but this is another really good technique. You actually look at their cell membranes and look at their fatty acid profiles, and you can actually tell a lot about a microbe just from its fatty acid profiles. And so, so these are some of the things that we're gonna, especially like the morphology, motility, metabolism, and fits. Those things you'll definitely get experience with in the lab and start to train your eye to that. But it's kind of challenging in the microbial realm because, like I said, they don't often look different from each other. And so, when you take an evolution class, you tend to learn about, well, a new species arises when it can't mate with an individual and produce viable offspring. That's usually how they say, well, now you have a distinct species. And so, that's fair. They call it the biological species concept. If you have a fancy way to say it is reproductive isolation, that just means you can't have those organisms produce viable offspring anymore. Well, then you would say, well, that's a new species. Well, okay, so what about if you have organisms that don't have sex? the way we define sex. And microbes don't have sex, poor things. And so they're left with this idea that, okay, how do we talk about this with microbes? How do we wrap our head around this? And so if you think about like sex in the human and animal world, it's this idea where you have to end up getting sperm and egg to unite. And so that has to go, that means some of the cells in your body go through meiosis to produce the sperm and the egg. And then you have to go through the sexual event to get the merging of the sperm and the egg. Microbes don't do that. They don't do that. So it's like, okay, so now how do we define speciation if they're not having sex? So this second bullet, I'd like you to memorize, please. This is a pretty well accepted idea of microbes version of sex. And this is proposed by Lynn Margulis from the endosymbiosis theory and her son, Dorian Sagan. And so they say, well, okay, if we define sex as the formation of individual organisms that contain genes from more than a single source or more than a single parent, then that's microbial sex. And that works. That works because microbes don't have egg and sperm. They don't do that. But they do this. They can contain genes from more than a single source or parent, so to speak. So another thing you need to appreciate about microbes, if they go, you know what, I have enough carbon and energy and electrons, I can go ahead and reproduce. They certainly aren't doing egg and sperm. All they do is faithfully, hopefully, copy their DNA, double everything in their cell, and split in two. That's pretty much it. And it's called binary fission. They just get a little bit bigger, make good copies of everything, and then split in two. And so then you have two progeny cells. And okay, so that would be their idea of vertical transmission, right? When you talk about vertical transmission of DNA, it's from parent to offspring, so to speak. And so you have the, you know, the original cell and then the two progeny. So that's vertical. But we're going to also consider horizontal transfer, which is a huge thing. So horizontal gene transfer. We know that coexisting microbes in the same, you know, physical proximity, they can share genes with each other. And they know there's several ways to do this, and they're discovering more ways now than what they thought. And when this was first discovered, it was done in a lab environment where you had like just like a bunch of E. coli maybe growing in a test tube or something. And all those individual cells of E. coli were floating around next to each other. And they could witness that one E. coli cell gave DNA to another E. coli cell, which was pretty big news. That was like, what? You know, that you have two coexisting microbes where you have some DNA exchange. And so they thought, well, that's really cool, but it's probably can only happen within individuals of the same species, like two E. coli hanging out with each other. Then they discovered that it is not just limited to that. You can have more unrelated now. So let's say between different species. So let's say you have, you're going to work with Pseudomonas fluorescens, and you're going to work with Pseudomonas aeruginosa. So those are two different Pseudomonas species. And they witnessed that Pseudomonas fluorescens could give DNA to Pseudomonas aeruginosa. And they're like, that's crazy because they're not even the same species. And so it was met with a lot of incredulity, if I'm saying that correctly. And then they said, what about different genera? So they did experiments with that. Sure enough, different genera of microbes were sharing DNA. And now they know even between different domains, you can have sharing of DNA. A popular example I like to give is plants and microbes. We know that microbes can insert some of their DNA right into the plant chromosome. And they can do some fabulous things for the microbe. Not so much for the plant, but you have this idea that really unrelated, domain is, super unrelated organisms compared to species. And yet you still have successful DNA transfer and expression of those genes. Okay, that's the part two of it. You have to get them expressed, of course. They have to get turned on and expressed for that to be like something, successful. So they started to say that microbes are promiscuous or cosmopolitan. They actually describe it that way. And it's true though. I mean, they're really good. And they, when they first started looking at this in the lab, they're like, oh, it's probably just like the organism they were currently studying. It's probably this organism's just so special. And then they realized, no, it's not just that organism. There's all kinds of microbes that are doing this. And so now they're almost flipped the other way where they say this is more commonly done than not done among the different organisms. And so this idea that you have really it's the microbe version of sex where they can have two living coexisting microbes hanging out in the same environment, and there can be horizontal transfer of DNA. Sometimes it's called lateral gene transfer, but that term has seemed to have gone by the wayside, and they talk more about horizontal gene transfer. And then, of course, like I said, vertical gene transfer is from parent to offspring. So you still certainly have that, but horizontal is something that was just kind of mind-blowing for the scientists at the time that were studying this. And so when you think about it, from a prokaryotic cell, they can certainly have mutations in their DNA. That's still happening. They can have during their reproductive process, or maybe some chemical caused a mutation in the DNA. So they can certainly have that. And if it is something that's making them better adaptive to their environment, well, yeah, then they get speciation could occur from that. But don't forget about the horizontal gene flow. You certainly have both DNA mutations going on, and you have horizontal gene flow. And so now, once they recognize that, they started thinking more about like that phylogenetic tree I just showed you. They're like, the way this is written, you have one line going off to the bacteria, one off to the eukarya, and then jumping off of that is the archaea. And they're like, but microbes, you remember protocells, they're microbes, they're, ancient prokaryotic cell types. If they're exchanging DNA, the tree should be a little bit more horizontal. Right? So they're trying to say, this is how we thought about it previously. We need to think a little bit more like the bottom image, where you have this horizontal transfer. All right. And so they're trying to show that you don't have such a clear-cut speciation event here. You have this flow. So I've talked with a lot of microbial ecologists about this, and several that I've talked to seems to be the kind of the general consensus that when you get into the microbial world, sometimes it can be really hard to say what is a species, species A versus species B, because there's so much of the horizontal stuff going on. It gets rather cloudy on like this is a distinct species from this one. And so there's a lot of interesting talk about that. Here's an image from another, not from your textbook, but again, just trying to show your phylogenetic tree of life is not so clear cut. You have all this horizontal transfer and interconnection going on. And so if you talk to an evolutionary microbiologist in particular, they'll love to talk to you about that more. And here, again, I like this idea too, where they're showing that with microbes, so they're showing two examples here, Escherichia coli and Salmonella enterica. And what I like about this image is that they are talking not only about gain, genes being gained. So you see all that happening, okay? But they're also talking about genes being lost. And I think that often gets overlooked in an intro evolution class, you have to think about the idea that some of these genes that are no longer providing any benefit for that organism, especially in the microbial world, they can purge it. It's just too much upkeep. So gain and loss is definitely a process that occurs in the microbial realm. And yes. Vertical transmission happens during binary fission. Yeah, happens during binary fission. So you're going to see, I don't know, 15, 20 different microbes in the lab over the semester probably. And the other thing I need to caution you about, and we try very hard to minimize this issue, but the microbes that we tend to have available for you are microbes that we buy from the company, we make sure they're pure, and then we freeze them for long-term storage, and then we just, thaw them out and grow them up and we work with them. It's cheaper that way, right? So, but that means that the microbes you'll be given are lab microbes. They've lived their life in a lab, which means they're adapting to their current environment, right? So you're gonna work with an E. coli that I've had and used for the last decade. Now, E. coli, when we grow it up, we freeze it and it goes to sleep. And then we thaw it out and pop it into some lovely broth that has everything it needs. We shake it so that it's getting this liquid all bathed around it, and then it's got plenty of oxygen because it likes oxygen, and we give it the sweet life. E. coli is supposed to be motile with flagella. But if you're getting shaken around in this broth and you're getting babied and you got everything you need, would you want to build a flagella? That's a lot of effort. You'll see pretty soon when we start talking about cell structure. So I've run into this problem where we want to show you a lab exercise to prove this microbe is motile, can move on its own versus non-motile. And once in a while, we end up with strains that are so comfortable with the life we've given them that they've decided not to form flagella. And then we run into false negatives. Drives me nuts. But so we try to minimize that. But again, just trying to point out that these types of exchanges of DNA, if it, they're not going to express them or even keep them if it's not favorable to their habitat. And so they can lose even the genes, maybe not even just turn off the genes, just purge them. If they got the sweet life, they're like, I don't need that structured DNA.
Are you able to tell what genes are lost if it's not like the living?
Well, essentially they're doing comparative DNA sequencing of the generations of microbes that they're growing up, essentially. I have a few things at the end of this slide that I wanted to mention. But basically, they're tracking the DNA sequences over time as they're working with these microbes, and then they can pinpoint which ones are lost. But it has to be done over time of growing these microbes. So it's a challenging thing at the microbial realm to really kind of pinpoint what is a species and is this a separate species from that one, and it can get a little confusing. So this is a table that's commonly provided to get you thinking about how do they differ or how are they similar? And this is done at the domain level. So the top portion of this table is showing all things in common, like all living organisms have this kind of stuff going on. But then down here, they start to say, okay, there are some differences. What are they? And so you have the 3 domains, and they're saying, well, bacteria and archaea have these traits in common, right? And then the eukarya differ from those two in having slightly different situations going on. So that's how you interpret that table. And this is another one from your book, and this is good too. But for ease of what I'm asking you to look at, I tend to use this table. So as you're studying for the exam content, I would like you to know the comparison of the three domains off of this table is probably easiest. If we go down to all the way down to ribosomes, so all the way down to there, I'd like you to know that content. And then I'd like you to know the plasmid comparison. Okay. So those will be fair doing for the exam questions. Okay, so all the way down through ribosomes. And all of that, and then the plasmid. And then. Yeah, we're good on that. You'll get some of this as we go through the class. But not for this first exam. So the next set of slides is taken from a different, well, I think it's from your textbook, but these are slides telling you a story about Dr. Lenski's grand experiment that go with this idea of the evolutionary process. And so I'd like you to read this. And I think it's talked about in the textbook as well. And be familiar with this because I will ask questions on it on the exam. But basically this is a demonstration of evolution in process in a microbiology lab. And so please read through that. And next week we'll chat about it a little bit in class. But one of our former students was on this project. He came here to do his master's and he worked on this project. So it was fun chatting with him about it. But this is, when I talk to non-scientists about the evolutionary process, it's very easy for me to say, well, it's happening all around you. I mean, microbes can do it in the lab during your lifetime. You don't have, like with animals, you got to wait and wait and wait for the evolutionary process. But with microbes, you can see it within a very short stretch of time. And so it's easy to show and talk about the evolutionary process when you use microbes as an example. So please read through those slides and you can look in the textbook as well. But I'd like to shift gears. Well, not really shift gears. I'd like to go into a little bit more now on horizontal gene transfer. And I'd like to get a little bit more into like, what is that process? And let me just introduce it before. So we have this idea that microbes that hang out living next to each other can exchange DNA. So here, what you're seeing in figure A is a Petri plate where microbes have been growing on it. So when I do these types of experiments, I usually put a biocontrol agent, I spot inoculate, it's called. You take a loop and you just spot inoculate a clump of your biocontrol agents onto the plate. And before you've done that, you spread, you're gonna learn the spread plate technique Tuesday. You use a spreader and you spread a suspension of your test organism across the plate. And then after that's soaked in, you spot inoculate your biocontrol agent. And what I'm interested in doing is incubating that plate and seeing if my biocontrol agent can inhibit or kill my test organism. So here, what you're looking at is after the incubation and the test organism has grown up, all this kind of gray is all the test organism. And then this big dark spot is the inhibition zone or the kill zone. So what's happened here is my biocontrol agent has grown and excreted out some kind of antibiotic or something that's killed all the test organism cells in this area. Does that make sense? But look what you see in that kill zone. What do you see in that kill zone? You see these little dots? These are some of the test organism cells that manage to withstand the antibiotic that this biocontrol agent is excreting. And so somehow it was able to withstand that. It could have been a mutation. Could have been a horizontal gene transfer event of neighboring cells. But it looks like at least 1, 2, 3, 4, 5, 6, 7, 8 cells that had landed there on the plate somehow gained resistance to that antibiotic being exuded out by the biocontrol agent. And so it could be a mutation, like I said, or it could have been a horizontal gene transfer event that allowed for those cells to become resistant to the antibiotics. And that is one of the big issues for us, that we really care about horizontal gene transfer, because I'm sure unless you've lived under a rock, you've heard about antibiotic resistance, right? It's a big deal, and it's only going to get bigger unless we solve this problem. And antibiotic resistance is by and large done through horizontal gene transfer. That's why if you go to the hospital environment where all those antibiotics are getting used in a very closed environment, you need to be very careful. When you get out of that hospital, you wash your hands very well. All right, because in a hospital setting, you have a perfect condition, all these microbes getting exposed to antibiotics. That means a lot of resistance in those microbes are building up. It's a hot spot for resistance to occur because they constantly are getting exposed by the antibiotics. So you should be very careful when you go to visit, or I hope you don't have to go there for your own, but maybe you're visiting a sick person. Wash your hands, because you don't want those microbes that have the antibiotic resistance coming with you, right? Okay, we'll stop there. Remember, we don't have class on Monday, but we will have lab on Tuesday. Okay, you're going to learn the streak and spread plate technique, huge important techniques. So have a good weekend.
What about? That hospitals have stronger. Stuff in the-
Mhm.
Oh, hospital has sick people.